So one of them, the most common ones that you probably have heard, especially for microarray or bulk RNA-seq analysis, is COMBAT. And COMBAT uses Bayes regression. So it's literally almost the same as a regression, as we have also mentioning the normalization step. So you can use the batch information and regress that from your data. And it works well in small and medium data sets, but it assumes that all data sets need to have a similar cell type. So if you have the exact same cellular composition and distribution, so for example, if you have a cluster that is big in dataset 1, that cluster should also have the same amount of cells, or at least a proportion in dataset 2. And that will perform perfectly fine. But if you have data sets that are not with the same cell composition, then COMBAT or any linear regression will not work for your data set. And this is the common case. Most of the cases, the data sets are not in the same cell proportion. So, and that is the same issue for if you do the remove batch effect with Lima, or if you try to remove batch effects using the scale data, or if you try to use COMBAT from the SVA package, or rescale batches with the Bachelor, which is a package for batch correction in single cell. So these do not account for differences in cell composition and assume that batches are additive. One comment on this one is that, for example, different cell types have different batch effects. So for example, if you have two data sets, and if you consider T cells, if you sequence them deeper, maybe you have, for example, more CD3, CD3E, CD3D. So these are markers for the T cells. So these are the genes that will drive the batch differences between the T cells in these two data sets. But if you consider, for example, the other cell type, B cells don't have any of those genes, but they will have different genes. When you try to do linear regression, you will try to find those genes as well. So you try to increase the amount of T genes on the B cells as well. And that is not true. So that's what it means, that yes, we cannot assume that the batches are additive. And one of the things that will be overprone to overcorrection, which means, for example, in the case of the B cells, trying to correct for T cell genes, because that is a gene that is shared in that batch. So that's why these regression-based methods don't work well for a single cell unless you have a very the same cell distribution or composition between the batches. One method that also already mentioned is ZIMWave. That is matrix factorization, just similar to what PCA is, but also accommodate the gene and cell covariates. So this is also similar to another method that is called ZIFA, which is literally zero inflated PCA almost. And it works quite well on simpler and small medium data sets, but just because it's a matrix factorization, it will be very slow on very big data sets. But basically it's the same concept as the PCA. So you have your matrix with genes and samples, and then it's a multiplication and addition of other matrices accounting for the gene-wide covariances, the sample level covariances, and the sample covariates as well, unknown and known covariates. But in general, you don't need to understand very into details, but the idea is just this is a matrix multiplication way of correcting for batch effects. SC merge is also, I think, also mentioned as well in the normalization.
Sorry, can I have a question?
Yes, absolutely.
So you're saying this is slow on large data sets, presumably because the data is, well, but very large. But can you, is it possible to do dimensional reduction before doing this? And then correct the dimensional reduced data?
Maybe, yes. I haven't tried it myself, but it should be possible as well, because the only thing that will change is this gene level covariates, but then you can just input this one to not be accounted for. I think so. You can try. Yeah.
All right.
I know that the result of the ZIM wave is a PCA. So if you do ZIM wave, the result is a denoised PCA. But I think it should be possible. And so then you basically start with the PCA and then result in a PCA as well. But that should reduce the computational time that you spend on it.
Yeah.
Yeah.
Okay. Well, good try. Thanks.
Another method is also SC merge, which is very similar to ZIM wave, but now we're not using all the genes to calculate how you should normalize the samples or avoid the batches, but it will now use what they call the single cell stably expressed genes. And those genes are usually housekeeping genes that you see. And then they created a list from the paper. And basically, they look for the expression of those genes across all the batches and assume that those genes should have the similar expression across all data sets. And as well, it works well on simpler and small, medium data sets, but it also will be slow on larger data sets because it needs to compute all the normalization factors for all cells and so on. And if I'm not wrong, it also outputs as a PCA.